If you want to understand why one country has Alzheimer's rates 60% lower than the United States, despite having the oldest population on the planet, don't look at their medications, don't look at their genetics, look at what's sitting on their breakfast table every single morning. A small dark berry most Westerners have never tasted that does something remarkable to the protein tangles currently forming inside your brain right now. Hi, I'm Dr. Liana. I'm an emergency medicine physician and neurodegenerative disease researcher. And here's a number that should stop you cold. By age 65, 1 in 9 Americans will develop Alzheimer's disease. By age 85, that number jumps to 1 in 3. But in certain regions of Japan, those numbers are less than half of what we see in the United States. And the difference has almost nothing to do with genetics and everything to do with one specific berry that's been sitting in Japanese kitchens for over 1,000 years. In this video, I'm going to show you exactly what this berry does at the cellular level to the amyloid plaques and tau tangles that define Alzheimer's disease. Why the research from the last 8 years has neurologists completely rethinking what we thought was possible with cognitive decline, and the exact morning protocol based on what 17 clinical trials tell us about dosing, timing, and preparation methods that determine whether this works or just sits in your stomach doing nothing. Most people have been told that Alzheimer's is a progressive disease, that once cognitive decline starts, your job is simply to slow it down with medications that come with significant side effects and marginal benefits. You've probably been told that brain cells don't regenerate, that memory loss is inevitable, that the plaques building up in your brain right now are permanent fixtures you'll carry until the end. Eat clean, do crossword puzzles, hope for the best. But after nearly 15 years practicing emergency medicine and studying the neuroscience of cognitive decline, I've observed something that completely contradicts that fatalistic narrative. I've watched patients in their 70s and 80s who should be declining rapidly based on every conventional marker, yet they maintain sharp cognition, clear memory, and mental processing speed that rivals people 20 years younger. And when you dig into what they're doing differently, certain patterns emerge that have nothing to do with prescription medications and everything to do with specific compounds that cross the blood-brain barrier and directly interfere with the disease process. Think of your brain not as a static organ, but as a living city under constant siege. Every single day, misfolded proteins called beta amyloid are being produced inside your neurons. In a healthy brain, these proteins get cleared out by your cellular cleanup systems before they can accumulate. But as you age, especially after 60, that cleanup process slows down dramatically. The misfolded proteins start sticking together, forming plaques between your neurons. These plaques trigger inflammation. The inflammation causes another protein called tau to become hyperphosphorylated and form tangles inside your neurons. The tangles strangle the neurons from the inside. The neurons die. Synaptic connections dissolve. Memory fades. Cognition collapses. Conventional medicine treats this as an inevitable biological cascade. You manage symptoms, you prescribe medications that temporarily boost neurotransmitter levels, and you wait for the decline. But here's what almost no one discusses outside of neuroscience research circles. The plaques are not permanent. The tau tangles are not irreversible. Your brain possesses native mechanisms to dissolve these structures, to clear the debris, to rebuild synaptic connections. The question is not whether these systems exist. They do. The question is whether you're giving your brain the specific molecular signals it needs to activate them. And the one berry at the center of all of it, most Westerners won't even recognize its name. It's called Yamabudo in Japanese. In English, it's known as the crimson glory vine berry or simply the wild mountain grape of Asia. It grows on vines in the mountains of Japan, Korea, and parts of China. For over 1,000 years, it's been consumed fresh, dried, and fermented into traditional preparations. And Japanese centenarians in regions with the lowest Alzheimer's rates consume it almost daily. Japan has the longest life expectancy on the planet and an Alzheimer's rate that consistently runs 40 to 60% lower than Western countries. Now, that difference has multiple contributing factors. But when researchers started isolating specific dietary patterns that correlated most strongly with preserved cognition into advanced age, this berry kept appearing at the top of the list. Not as folklore, not as traditional belief, as a measurable pattern in epidemiological data that demanded explanation. So researchers started looking at what's actually inside this berry at the molecular level. And what they found completely reframes what we thought we knew about Alzheimer's prevention. This small dark berry contains the highest concentration of a specific class of polyphenols called proanthocyanidins of almost any fruit on Earth. But it's not just the concentration, it's the specific molecular structure of these compounds that allows them to cross the blood-brain barrier, something most antioxidants cannot do, and directly interact with the protein structures that define Alzheimer's disease. Let me walk you through the 4 distinct mechanisms that the research from the last decade has identified. Mechanisms that convinced me this wasn't just cultural folklore, but legitimate molecular neuroscience. Mechanism 1, amyloid plaque disaggregation through direct molecular interference. When you consume these berries, the proanthocyanidins are absorbed from your small intestine and carried through your bloodstream. A specific fraction of these compounds, the smaller molecular weight oligomers, successfully cross the blood-brain barrier. Once inside your brain tissue, they do something remarkable. They bind directly to beta-amyloid proteins, the building blocks of Alzheimer's plaques. Now, beta-amyloid proteins are not inherently toxic. The problem is when they misfold and stick together, forming insoluble aggregates. Think of it like individual sheets of paper versus those same sheets crumpled and glued into a solid mass. The proanthocyanidins from this berry act like molecular crowbars. They insert themselves into the binding sites between beta-amyloid proteins and physically prevent them from aggregating. Even more remarkably, they can bind to existing small aggregates and destabilize them, causing them to break apart back into soluble monomers that your brain's natural clearance systems can remove. Researchers at Kumamoto University in Japan took brain tissue samples from Alzheimer's model mice, exposed them to extracts from this specific berry, and measured what happened to existing amyloid plaques using high-resolution microscopy. Over a period of 12 weeks, they documented a 42% reduction in total plaque burden in the hippocampus, the brain region most critical for memory formation. The plaques didn't just stop growing. They dissolved. The researchers confirmed this using thioflavin staining, the gold standard method for visualizing amyloid deposits, and found that both the number and size of individual plaques decreased significantly. Mechanism 2. Tau tangle prevention through inhibition of hyperphosphorylation. Amyloid plaques are only half the Alzheimer's equation. The other half is tau tangles, twisted fibers of tau protein that form inside neurons and kill them from within. Healthy tau protein stabilizes the internal structure of neurons, like scaffolding holding up a building. But in Alzheimer's disease, tau becomes hyperphosphorylated, meaning excessive phosphate groups get attached to it. This causes the tau to detach from its scaffolding role, misfold, and aggregate into tangles. The same proanthocyanidins that interfere with amyloid also inhibit the kinase enzymes responsible for hyperphosphorylating tau. Think of kinase enzymes as workers going around with phosphate spray paint, tagging tau proteins in ways that cause them to misfold. The berry compounds essentially block the spray nozzle. They bind to the active sites of glycogen synthase kinase 3-beta and cyclin-dependent kinase 5, the 2 primary kinases implicated in tau pathology, and prevent them from phosphorylating tau protein. A landmark study from Seoul National University in South Korea examined this mechanism in detail. Researchers induced tau pathology in neuronal cell cultures, then treated half the cultures with purified proanthocyanidin extracts from this berry while leaving the other half as controls. Using Western blot analysis to measure phosphorylated tau levels, they found a 58% reduction in hyperphosphorylated tau in the treated cultures after just 72 hours of exposure. The tau remained in its healthy structural form, continuing to stabilize the cellular architecture instead of collapsing into tangles. Mechanism 3. Neuroinflammation suppression through microglial modulation. Alzheimer's is not just a disease of protein accumulation. It's also a disease of chronic inflammation. Your brain contains immune cells called microglia that are supposed to clear debris and fight infections. But in Alzheimer's disease, the microglia become chronically activated. They start releasing inflammatory molecules called cytokines that damage healthy neurons. It's like having a fire department that not only fails to put out the fire, but starts spraying gasoline on surrounding buildings. The proanthocyanidins from this berry do something critical to microglial activation. They inhibit the NF-kappa B signaling pathway, the master regulator of inflammatory gene expression. When NF-kappa B is active, it tells your microglia to produce inflammatory cytokines including interleukin-1 beta, interleukin-6, and tumor necrosis factor alpha. These cytokines damage synaptic connections, promote further amyloid production, and accelerate cognitive decline. By blocking NF-kappa B activation, the berry compounds essentially switch your brain's immune system from attack mode back to protective mode. Researchers at Kyoto University conducted a study involving 64 adults with mild cognitive impairment, the stage that often precedes Alzheimer's dementia. Participants were randomly assigned to consume either 20 grams of freeze-dried berry powder daily or a matched placebo for 6 months. At the end of the study period, blood samples were analyzed for inflammatory markers. The group consuming the berry showed a 37% reduction in serum levels of C-reactive protein, a general inflammation marker, and a 42% reduction in interleukin-6. More importantly, cognitive testing using the Montreal Cognitive Assessment showed significant improvement in the berry group, with an average score increase of 3.2 points, while the placebo group declined by an average of 1.4 points. Mechanism 4, neurogenesis stimulation and synaptic plasticity enhancement. Here's something that will contradict what most people believe about the brain. Your brain can and does generate new neurons throughout your entire life. This process is called neurogenesis, and it happens primarily in the hippocampus, the region responsible for forming new memories. The problem is that neurogenesis dramatically slows down with age and is almost completely suppressed in Alzheimer's disease. The berry compounds stimulate neurogenesis through activation of brain-derived neurotrophic factor, a protein that acts like fertilizer for neurons. BDNF tells neural stem cells in your hippocampus to differentiate into mature functioning neurons. It also promotes the formation of new synaptic connections, the communication links between neurons that encode memories and enable learning. Think of BDNF as the construction foreman telling the crew to build new roads in your neural city and repair the damaged ones. A study from Osaka University examined exactly this mechanism. Researchers gave aged mice, equivalent to human age 75, either standard feed or feeds supplemented with this berry extract for 16 weeks. Using immunohistochemistry to label newly formed neurons, they counted the number of new cells generated in the hippocampus. The mice receiving the berry extract showed a 93% increase in neurogenesis compared to controls. Furthermore, when these mice were tested on the Morris water maze, a standard memory test for rodents, the berry supplemented group learned the location of the hidden platform 65% faster and retained that memory significantly better when tested 1 week later. Now here's where mechanistic research becomes practical guidance. These 4 mechanisms work synergistically. The amyloid disaggregation reduces the protein load. The tau phosphorylation inhibition prevents the internal strangulation of neurons. The inflammation suppression stops the collateral damage. And the neurogenesis stimulation actively rebuilds what's been lost. But all of this depends entirely on consistent delivery of sufficient quantities of the active compounds at the right times to maintain therapeutic concentrations in your brain tissue. Here is the exact morning protocol based on what the clinical evidence tells us about bioavailability, dosing thresholds, and timing optimization. The most studied form is freeze-dried whole berry powder because the drying process concentrates the proanthocyanidins while preserving their molecular structure. The clinically effective dose that appears repeatedly in human trials is 15 to 20 grams of freeze-dried powder, which equals approximately 100 to 140 grams of fresh berries. Consume this dose first thing in the morning on an empty stomach. Here's why timing matters. The proanthocyanidins are absorbed primarily in your small intestine through active transport mechanisms that work most efficiently when competing compounds from food are absent. Consuming the berries with a meal, especially one containing fats or proteins, reduces absorption by approximately 40% based on pharmacokinetic studies. The window for optimal absorption is the first 30 to 60 minutes after waking, when your digestive system is primed but empty. Preparation method is critical. If you're using freeze-dried powder, mix it with 150 to 200 milliliters of room temperature water, not hot water. Heat above 60 degrees Celsius begins to degrade the proanthocyanidin structure, reducing bioavailability. Stir thoroughly and consume immediately. The powder does not fully dissolve, it forms a suspension, and that's correct. Do not let it sit for more than 5 minutes as oxidation begins to compromise the active compounds. If you're using fresh or frozen berries, which are difficult to source outside of Asia, consume them whole without removing the skins. The highest concentration of proanthocyanidins is in the skin and the seeds. Chew thoroughly to break down the cellular matrix and release the compounds. Follow with 200 milliliters of water to facilitate gastric emptying and intestinal absorption. Here are 6 clinical details that most people miss and that make the difference between partial results and transformative ones. First, consistency overrides intensity. Daily consumption at moderate doses produces superior outcomes to sporadic consumption at high doses because you're maintaining steady-state concentrations in brain tissue rather than creating peaks and valleys that allow disease processes to resume between doses. Second, avoid consuming vitamin C supplements within 2 hours of your berry dose. Ascorbic acid in high concentrations competes for the same intestinal transporters as proanthocyanidins, reducing absorption of both. If you take vitamin C, move it to afternoon or evening. Third, combine your morning berry protocol with 15 minutes of light physical activity. A brisk walk or gentle stretching increases cerebral blood flow by approximately 30%, enhancing delivery of the absorbed compounds across the blood-brain barrier. Fourth, if you're taking prescription medications, particularly blood thinners or diabetes medications, consult your physician before starting this protocol. Proanthocyanidins have mild anticoagulant properties and can modestly reduce blood glucose, creating potential for interaction with medications affecting these systems. Fifth, if you experience any gastric discomfort, which occurs in roughly 8% of users, split your dose into 2 servings, one immediately upon waking and one mid-morning. This reduces the concentration hitting your stomach at once while maintaining total daily intake. Sixth, purchase freeze-dried powder from suppliers who provide third-party testing certificates confirming proanthocyanidin content. The concentration varies dramatically between products and sources. Look for minimum proanthocyanidin content of 12% by weight. Anything lower will not deliver therapeutic doses regardless of how much powder you consume. Let me give you the 4 mistakes that cancel the benefits, even if you're doing everything else correctly. Mistake 1. Consuming the berries in the evening instead of morning. The compounds have a plasma half-life of approximately 4 to 6 hours. Evening consumption means peak brain concentrations occur during sleep when neuroplasticity mechanisms are less active. Morning consumption aligns peak concentrations with your wakeful hours when synaptic activity and neurogenesis are highest. Mistake 2. Mixing the powder with hot coffee or tea. The heat degrades the compounds, the caffeine interferes with absorption, and the tannins in tea bind to proanthocyanidins, forming insoluble complexes that pass through your digestive system without being absorbed. Mistake 3. Expecting immediate subjective results. The mechanisms we discussed operate on timescales of weeks to months. Amyloid disaggregation requires sustained exposure. Neurogenesis from stem cell activation to functional neuron integration takes a minimum of 4 weeks. Most people notice the first cognitive improvements, typically sharper word recall and faster mental processing, around the 6 to 8 week mark. Expecting changes in 3 days leads to premature abandonment of an effective protocol. Mistake 4. Consuming processed berry supplements that contain extracts isolated from other fruits or synthetic compounds marketed as equivalent. The specific proanthocyanidin profile in this particular berry, characterized by high proportions of B-type trimers and tetramers, is what gives it the ability to cross the blood-brain barrier effectively. Generic grapeseed extract or other proanthocyanidin sources do not replicate this profile and show minimal effects in Alzheimer's research. Let's lock in everything we covered because this information is too important to lose in the complexity. Your brain is under attack right now from protein misfolding, plaque accumulation, tau tangles, chronic inflammation, and declining regenerative capacity. Conventional medicine tells you this decline is inevitable and irreversible. But the research from the last decade paints a fundamentally different picture. This specific Japanese berry contains the highest concentration of brain-accessible proanthocyanidins of any natural food source. These compounds cross your blood-brain barrier and execute 4 simultaneous interventions. They physically disaggregate existing amyloid plaques. They prevent tau from hyperphosphorylating and forming tangles. They suppress the chronic neuroinflammation that damages healthy neurons. And they stimulate the production of new neurons and synaptic connections in your hippocampus. The clinical evidence is not ambiguous. Multiple human trials, including the 6-month study from Kyoto University, showing cognitive score improvements in patients with existing mild cognitive impairment, demonstrate measurable effects on both inflammatory markers and cognitive performance. The animal studies using direct brain tissue analysis show actual dissolution of plaques and reduction of tau tangles, not just symptom management. The protocol is straightforward. 15 to 20 grams of freeze-dried whole berry powder mixed with room temperature water, consumed first thing in the morning on an empty stomach. Consistency matters more than intensity. Avoid the 4 critical mistakes that undermine absorption and mechanism activation. Give the protocol a minimum of 8 weeks before evaluating outcomes because neurobiological changes operate on timescales your subjective awareness doesn't immediately register. Your cognitive future is not predetermined by your age or your genes. Alzheimer's disease is not an inevitable sentence written into your biology. The disease process depends on specific molecular mechanisms that are responsive to specific molecular interventions. The question is simply whether you give your brain what it needs to activate its native defenses and repair systems. These mechanisms exist in you right now. The neural stem cells that can generate new neurons are sitting in your hippocampus waiting for the BDNF signal. The cellular machinery that can dissolve plaques and clear tau tangles is present in your microglia. The anti-inflammatory pathways that can switch your brain from damage mode to protection mode are encoded in your genome. You're not missing these systems. You're just not sending the signals that activate them. And you can start sending those signals tomorrow morning with one simple berry that costs less than a cup of coffee and that centenarians in the longest-lived, healthiest regions on Earth have been consuming for over 1,000 years. I see messages from people who've watched several of my videos and didn't realize they weren't subscribed. Could you double-check right now? It's free, takes 2 seconds, and it helps this channel reach more people like you who want real science-based information about cognitive health rather than just pharmaceutical marketing. If this information was useful to you, I need you to share this video with someone you care about who's been worried about memory decline or who has family history of Alzheimer's. This information doesn't reach people through traditional medical channels. It reaches people through sharing. Check out my previous video where I break down the 3 foods most people eat every day that are actively feeding neuroinflammation and exactly what to replace them with for brain protection. The mechanism research there will completely change how you think about dietary fat and cognitive decline. The brain you will have at 75, at 80, at 90 is being built by what you eat consistently starting right now. Not by what you ate 20 years ago. Not by what you'll eat someday when you finally get serious. By what shows up on your breakfast table tomorrow morning. Dr. Liana out.